Hello, so my name is Kristen Fiore, and I'm a member of the Peterson Lab. And in this publication, we join forces with the Cheng Lab at the National Taiwan University, as well as the Chatterjee Lab at the Indian Institute of Science to study the structural impact of incorporating thioamides into a beta-hairpin. So for this study, we used two beta-hairpin scaffolds. We used the Gelman YKL, where there is a diagonal cation-pion interaction, and the less stable was the HPT, where we changed out the tyrosine to a threonine. And so we incorporated thioamides at all of the colored positions shown, and we wanted to study this with NMR. And so in order to do that, for each thioamide position of interest, we synthesized a corresponding folded and unfolded control. For the folded control, we added terminal cysteines and oxidized them to form a disulfide link macrocycle. And for the unfolded control, we switched the stereochemistry of the proline and the beta turn from D to L. And this allowed us to perform chemical shift deviation analysis, as well as to calculate the percent fraction folded, as well as the free energy of folding. And so one result that was relatively unexpected was for the HPT scaffold, when we incorporated a thioamide at isoleucine 10, which is an internal location, we observed that the fraction folded percentage was relatively similar to our oxoamide parent peptide, or the HPT scaffold. And so to further understand structurally what was going on, we took our experimentally derived distance constraints from the NMR, as well as a thioamide patch that our lab has previously published on, in order to structurally model the folded controls with Rosetta. And so, for example, for the isoleucine position, we were able to observe that due to a right-hand twist of the scaffold, the thiocarbonyl is actually solvent exposed, and this probably explains why it's a neutral position for incorporation. And so from the structural modeling, we were able to make some sense of some of the trends that we observed in the fraction folded control, but we additionally have more research to do to, for example, understand how the thioamide affects the conformational rigidity of the scaffold, as well as eventually our final goal is to develop a model to predict where a thioamide is structurally stabilizing, neutral, or destabilizing based on a peptide or a protein structure. And so with that, I hope you're interested to go and look at the paper, but otherwise, thank you so much for listening.